Why is epigenetic control of the DUX4 gene critical in FSHD? Imagine your genes as switches that turn on and off to control how your body works. In facioscapulohumeral muscular dystrophy, or FSHD, one particular gene called DUX4 becomes a problematic switch that stays turned on when it should be off. This is where epigenetics, the system that controls gene activity without changing the actual DNA sequence, becomes so important. Normally, the DUX4 gene is kept silent in mature muscle cells by epigenetic mechanisms. These include DNA methylation, which adds chemical tags to the DNA to prevent gene activity, and repressive histone marks that make the chromatin structure tightly packed. This tight packing prevents the gene from being transcribed into messenger RNA, so the DUX4 protein isn't produced in healthy muscle tissue. DUX4 is usually only active during early embryonic development or in the testis, not in adult muscles. In FSHD, this carefully maintained epigenetic silencing breaks down. The disease often results from a contraction of the D/Z for repeat region on chromosome 4. This contraction causes the chromatin to relax, losing those DNA methylation marks and repressive histone modifications. As a result, the chromatin becomes more open, allowing the DUX4 gene to be abnormally expressed in skeletal muscle cells. Sometimes, mutations in genes that regulate epigenetic modifications also lead to this relaxation, even without repeat contraction. When DUX4 is misexpressed in muscle cells, it acts like a rogue transcription factor. It activates genes that are normally only turned on during early development, which can be toxic in adult muscle tissue. This toxic activity causes muscle cells to undergo apoptosis or programmed cell death and disrupts normal muscle function. DUX4 also interferes with muscle regeneration by opposing factors like PAX7, which are essential for muscle repair. The result is progressive muscle weakness and degeneration, characteristic of FSHD. Interestingly, once DUX4 is expressed, it can inhibit the cell's natural mechanisms for degrading its own mRNA, such as nonsense-mediated decay. This stabilization of DUX4 mRNA amplifies its harmful effects, creating a vicious cycle that worsens muscle damage over time. Because the root cause of FSHD involves the failure to epigenetically repress DUX4, scientists are exploring ways to restore this repression. Potential therapies aim to modify the epigenetic landscape, encouraging the chromatin to become more compact and silencing DUX4 again. This approach could slow or stop the progression of muscle degeneration. In essence, controlling the epigenetic state of the DUX4 gene is crucial because it prevents the gene from being turned on when it shouldn't be. When this control is lost, DUX4's toxic effects take over, leading to the muscle weakness seen in FSHD. Understanding this process helps researchers develop targeted treatments that could one day restore proper gene regulation and improve quality of life for those affected. Always remember, for any health concerns or treatment options, consulting a healthcare professional is essential to get personalized advice based on the latest scientific understanding.